Hello, dear first secondary students. Today we are going to revise our syllabus for the second term. Wish you a good luck, inshallah. Let's start by our first unit, genetics. First lesson, chromosomes and genetic information. As we know from our previous studies that chromosomes are exist inside the nucleus. Nucleus exist inside the cell of all living organisms. The structure of the nucleus, generally it consists of two parts. They are two chromatids connected from the center by a centromere. This is the general structure of the chromosome. While the chemical structure of the chromosome consists of two chemical substances, which are DNA and proteins. DNA and proteins. And this is the chemical structure of the chromosome. As we studied before in our first term, that DNA is a polymer that consists of smaller units which are called monomers. What if you remember the name of the monomer of DNA, which is nucleotide? So group of nucleotides together or a sequence of nucleotides is called a gene. What's meant by the gene? Gene, it's the part of DNA which is responsible for the appearance of a special genetic trait by producing an enzyme which is necessary to complete a reaction to form a special type of protein. If this protein exists inside your body, that trait will appear. So gene is an important part which is responsible for our hereditary traits. And this gene exists in the DNA, which exists inside the chromosomes. As we know from our previous studies, there are two types of cells, somatic cells and reproductive cells or gametes. What is the difference between somatic cells and reproductive cells? Somatic cells, they are all the body cells, the cells of all our body except cells of reproductive organs or gonads. So all our body cells are called somatic cells. The cells of reproductive organs are called reproductive cells or gametes. Cells of our body like skin, muscles, blood, nerve cells, all of our body cells. These cells undergo a special type of cell division which is called mitotic cell division or mitosis. This type of division produces a cell copy from the parent cell. And these cells contains total number of chromosomes, as we know before from our previous studies that each organism has a constant, a fixed number of chromosomes. Like in the human cells, we have 46 chromosomes. In rabbit cells, somatic cells, we have 44 chromosomes. This number is called diploid number of chromosomes or total number. While inside the reproductive cells or gametes, these cells undergo another type of cell division, which is called meiosis or meiotic cell division. This division produces cells which are called gametes, and these cells contain haploid number of chromosomes or half number, which is called N chromosomes, like cells of testes and ovaries to produce sperms and ovum, or to produce pollen grains in case of plant cells. Now we are going to know one from the most important topics in our syllabus, which is called karyotype. What's meant by the karyotype? Karyotype means the arrangement of our chromosomes inside the cells in a descending order according to their size, So our chromosomes inside our bodies as human or inside the body of any living organisms, chromosomes are arranged in a descending order according to size. For example, inside our body cells, we have 46 chromosomes or 23 pairs of chromosomes. These chromosomes are arranged in the form of homologous pairs, similar pairs, identical, symmetric. They are homologous pairs arranged in the form of, or arranged in a descending order according to their size. Okay? We have two different types of cells, cells for male and cells for female. The karyotype for male consists of 22 somatic cells or 22 autosomes and 2 sex chromosomes. And the cells of female, or the karyotype of female cells, consists of 22 pairs of chromosomes, which are called autosomes, and 2 sex chromosomes. Now, let's differentiate between the sex chromosomes in male cells and female cells. Sex chromosomes in male cells are asymmetric or heterozygous. They are different chromosomes, X chromosome and Y chromosome. While in the female cells, the sex chromosomes are identical. They are symmetric, 2 X chromosomes. So inside the nucleus of our cells as a human, our somatic cells, we have 46 chromosomes that are arranged in a descending order in the form of, according to size, in the form of a homologous pairs. So in our body's somatic cells, we have 23 pairs of chromosomes. While each gamete contains how many chromosomes? Only 23 chromosomes. So in the gamete cells, the chromosomes are arranged in the form of pairs or single chromosomes. They will be single chromosomes. So we are going to differentiate between somatic cells and reproductive cells according to the number of chromosomes. If the karyotype consists of chromosomes that are arranged in pairs, so this is a karyotype for a somatic cell. If these chromosomes are arranged in the form of single chromosomes, these cells will be gametes. Okay? So chromosomes, pairs, in case of somatic cells, are arranged in a descending order according to size from number 1 to 23. From number 1 to 22, as we know before, they are called somatic cells or autosomes, somatic chromosomes, sorry, or autosomes. While chromosome pair number 23 is called sex chromosome, which is different from the male cell than female cell. But we need to know that the normal arrangement of the sex chromosomes according to size is the 7th chromosome or the 8th chromosome, sorry. 8th chromosomes يعني after the 7th chromosome. This is the normal arrangement of the sex chromosomes according to size. But because they are different from the rest of autosomes, they are put in the last order, or they are considered as chromosome pair number 23 to be differentiated from the others because these two chromosomes, or this pair of chromosomes carrying the sexual genes only, while the rest of the autosomes carrying the somatic genes or the genes that are responsible for somatic traits. Okay? Okay. Now, let's start by our second topic, which is called Mendelian cases. I think that we studied this part before in the last year, third prep, but let's remember together what is called the Mendelian cases. Firstly, the first scientist who discovered or who talks about the inheritance of genetic traits, he is Gregor Mendel. Mendel was the first scientist who discovered the inheritance of genetic traits through different generations or through many generations by his experiments on the pea plant. He was the first one who may talk about this, the inheritance or genetics in general. Mendel makes two rules or two laws to explain the inheritance of genetic traits through different generations. First law, called law of segregation of factors. What's meant by the law of segregation of factors? From the most important conclusions for Mendel, that each genetic trait is controlled by two genes or two factors or two alleles. These genes are represented by letters. If these genes or these traits are, we have two types of traits. They are dominant traits and recessive traits. What is the difference between dominant and recessive traits? Dominant trait it's the strong trait. If this trait is present, the recessive trait will disappear totally, but in the first generation only. Then it will appear again in the second generation by ratio 25% or quarter. So if the trait is a dominant trait, it will be represented by capital letters. The genes which are responsible for this trait will be represented by capital letters. If this trait is a recessive trait, the genes representing these traits will be small. This trait will be small letters. So dominant traits may be, or each trait, as we said before, controlled by two genes, one from the mother and one from the father. So if the trait is considered as a dominant trait, the genes controlling this trait will be represented by capital letters. For example, A capital, A capital. So this trait, which is represent the yellow color in the pea seeds, is a dominant trait. But what about the recessive trait? The recessive trait must be represented by the same letter but in a lowercase or small letters, which is A small, A small. Why? Because this green seeds, or this trait, is the recessive trait for yellow seeds, which is considered as a dominant. But in case of dominant traits, we have two possibilities. This dominant trait may be pure dominant, so it will be represented by two capital letters, A capital, because it's a pure dominant, yani the genes from mother and father are dominant genes. And maybe it will be a hybrid trait, or A capital, A small. A is a dominant gene, A capital, A small is a recessive gene. Which trait will be represented by this genotype? It will be the dominant trait, but in a hybrid state. So in the first law, or law of segregation of factors, Mendel said that when two pure members that differ in any pair of allelomorphic characteristics, allelomorphic or contrasting traits means the dominant trait and its recessive. The dominant trait will appear in the first generation by 100%. Only dominant trait will appear in the first generation by 100%. While in the second generation, A capital, A small, crossing by A capital, A small, yani two hybrid yellow seeds, the product will be 75% yellow, dominant trait, and 25% green, which is considered as the recessive trait. Or the ratio between dominant and recessive in the second generation, in case of Mendel's first law, will be 3:1. So what about Mendel's second law? Mendel's second law, which is called law of independent assortment, it's to prove that each trait is inherited independently without being affected by the other. Yani if the plant or the seed of the pea plant is yellow seed or green seed as the seed color, and smooth or wrinkled seed as the seed shape, yani we are going to explain two traits together to prove that each trait from these two traits will be inherited independently without being affected by the other one. Okay, so after crossing between smooth and yellow colored seeds with green and wrinkled colored seeds, in the first generation, all the individuals will be yellow and smooth. يعني 100% dominance, which is called complete dominance in the first generation. When two individuals carrying the genotype Y capital Y small or capital R small will being crossed together, the product will be 16 individuals. These individuals, 9 of these individuals carrying the smooth and yellow color seed, يعني the dominant trait. One individual carrying green and wrinkled seed, يعني recessive traits, and 3 from these individuals are green but smooth, and 3 other individuals will be yellow but wrinkled, which proves that each trait will be inherited independently without being affected by the other trait. And this is which is called Mendelian cases. All the genetic traits that follow the Mendelian rules are called Mendelian cases. But there are some genetic traits that cannot follow the Mendelian rules. They are exceptions. These traits are called non-Mendelian cases. In our syllabus, we have some examples for these non-Mendelian cases. So let's start by the first non-Mendelian case in our syllabus, which is called lack of dominance. What's meant by lack of dominance? Lack of dominance means no dominance. Yani there is no dominant trait and recessive trait. No trait dominates over the other. The most important, the most obvious example in our syllabus is the flowers, the color of the flower in the Antrinium plant. This plant has 2 flower colors, which are red and white. As we studied in Mendel, if red and white, yani dominant and recessive, if they are yani dominant and recessive, are crossed together, the product will be dominant by 100% in the first generation. But when the scientists crossed the 2 plants or the 2 flowers of Antrinium plants, the product will be an intermediate trait. Not red flowers are produced or white flowers are produced, but pink flowers are produced. Why? Because there is no dominancy for the red on the white flowers or white on the red flowers. Each one of these traits are strong enough to express itself. So the product is a new trait, an intermediate trait between the 2 traits of the parents, which is pink color. So if we need to represent the traits, the genes for this case, the genes will be represented by only capital letters. There is no small letters. Why? Because there is no dominancy, lack of dominance, no dominant trait, no recessive trait. So red flower, but this is a special case. For example, in our Antrinium plant, red flowers are capital R capital, white flowers W capital W capital. So the product will be RW, which is represents the pink color. What about the second generation? When 2 pink colors are crossed together, the product will be red, pink, and white flowers by ratio 1 to 2 to 1. 1 for each trait from the traits of the parents, and 2 for the new intermediate trait. And this is which is called lack of dominance, no dominancy. When these 2 traits are crossed together, the product is a new trait. Okay, so both of the 2 genes represented by capital letters, as I said, because of the lack of dominance between these 2 genes. Now, you have to concentrate in this point that in the case of lack of dominance, genotype indicates the phenotype. Let's remember what's meant by phenotype and genotype. Phenotype, it's the observable trait. Yani if I said that this flower is red, can you observe the color of the flower? Yes, it's a red flower. This one is a white flower. So phenotype is the observable trait. While what about the genotype? It's the genetic structure, the structure of the genes of each trait. So red is considered red and white. They are the phenotypes, while R capital, W capital, W capital, R capital, W capital, this is the genotype. Here, if I said that the flower of Anthraeum plant is red, can you guess its genotype? Sure. Red flower, in case of Anthraeum plant, its genotype will be only R capital, R capital. There is no other possibilities. What about the white plant of the white flower? It will be WW. There is no other possibilities. Pink flower, R capital, W capital. It's the only genotype for this phenotype. So in case of lack of dominance, genotypes indicates the phenotypes. And this is our first case in the non-Mendelian cases. And here, a comparison between complete dominance or Mendelian cases and lack of dominance. Okay, let's start by the second non-Mendelian cases, which is complementary genes and lethal genes. There are two different cases. What's meant by the complementary genes? Complementary genes is considered as a genetic case in which the trait, the single trait, is controlled by two pairs of genes or 4 genes. This trait, or the dominant trait to appear, it's a must to the genotype must be at least has one dominant gene from each pair to appear or to be exist. Why? Because each of the two pairs of genes controls a certain enzyme, production of a certain enzyme. To complete a special reaction, this reaction is necessary to appear our dominant trait. So if we have, for example, this genotype, A capital, A small, B capital, B small. Here we have how many genes? 4 genes or 2 pairs of genes. Each pair is responsible for producing a certain enzyme. Each single pair is responsible for producing a certain enzyme to complete a special reaction which is necessary to appear our dominant trait. So if the pair number 1 contains at least one dominant gene, the reaction or the enzyme will be produced and the reaction will take place. If the second pair contains at least only one dominant gene, the second enzyme will be produced and the second reaction will be completed to form to appear the dominant gene. So this case is called complementary genes. Let's start to discuss some genotypes to differentiate between the genotype for the dominant trait and the genotypes for recessive traits. Here we have 2 white flowers, A capital, B small, B small. Let's start by our first pair. In the first pair, we have 2 dominant genes. Okay. In the second pair, there is no dominant gene. So the reaction will not completed and the pink color will not appear. So the color of this flower will be white flowers, which is the recessive color or the recessive trait. In the second parent, A small. In the first pair, no dominant genes. So the first enzyme will not produce and the first reaction will not occur. The second pair, okay, it has 2 dominant genes, but the reaction will not be completed. So the color of the flower will be the recessive one, which is the white flowers. But here, A capital, A small, B capital, B small. A capital, A small, yani the first pair has 1 dominant gene, so the first reaction will take place. the second pair has one dominant gene, so the second reaction will take place, and the reaction which is necessary to appear the dominant trait will be completed. Okay, this is which is called complementary genes. And this is the second non-Mendelian case. Let's start by our third non-Mendelian case, which is called lethal genes. What about the lethal genes? What's meant by lethal genes? These genes control some characteristics when they are found in a pure or homozygous state. This state may be dominant or recessive, and it leads to disruption of some vital processes, leading to death of almost 25% of the offsprings. So lethal genes, they are the genes that lead to death, but if they are excessed in a pure or homozygous state. This state may be dominant or recessive. When the genes are excessed in a dominant state, this trait is called, or this case is called, dominant lethal genes. If this trait is excessed in the form of pure recessive genes, this trait is called recessive lethal genes. In our syllabus, we have two examples for the dominant lethal genes, which are the yellow fur in mice and the bulldog strain in kettles. And recessive lethal genes, we have two other examples, which are absence of chlorophyll in case of corn plant and the infantile dementia in human. And these are our examples. What about the dominant lethal genes? If we have two yellow fur mice are crossed together, their genotypes Y capital Y small, Y capital Y small, gametes Y capital Y small, Y capital Y small, the products will be Y capital Y capital. And here is our case. Y capital means pure dominant genes, and this gene is a lethal gene. So this individual will die inside the mother's uterus before laboring. But the rest of the offspring of the rest of the individuals, Y capital Y small, they are yellow mice, and Y small, it's the recessive trait or the gray one. What about the recessive lethal genes? C capital C small, it's a green corn plant. C capital C small, it's a green corn plant. By the crossing between these two hybrid green corn plants, the products will be C capital, which is a green. There is no problem in this individual because we are in the case of recessive lethal genes. So the gene which leads to death is the recessive one, not the dominant one. C capital C small, it's hybrid. No problem in this part, in these two individuals. C small, it's the individual that will die. Why? Because this individual carrying the recessive lethal genes. And this is our third non-Mendelian case. Our fourth, before starting the fourth non-Mendelian case, we have to know that there are some environmental conditions affecting on the action of some genes. Like some environmental conditions, like air pollutants, lack of oxygen or oxygen deficiency, exposure to radiation, some environmental factors like light and temperature. As example, the gene which is responsible for the appearance of chlorophyll. This gene must be activated by sunlight, like the cabbage leaves. The cabbage leaves, the external leaves are green, and the internal leaves are white. Why? Because external leaves are exposed to the sunlight, so the gene which is responsible for the appearance of chlorophyll is activated, so the leaves are green. While the internal leaves, they are not exposed to the sunlight, so the gene is not activated, so they are white leaves. So if these leaves, these internal white leaves, are exposed to the sunlight, their color may be changed to be green. Yes, their color will be changed into green color, because in this case, the sunlight will activate the gene which is responsible for the appearance of chlorophyll. So we need to know that some genes are affected by some environmental conditions. Now, let's start by one from the most important parts in our syllabus, which is the inheritance of blood groups in humans. I think that all of you know that we have 4 different types of blood groups: blood group A, blood group B, blood group AB, and blood group O. We need to study for this 4 blood groups genetic classification and the chemical classification. So let's start by the genetic classification of blood groups. Blood groups genetic classification is controlled by 3 different genetic cases, which are complete dominance. What's meant by the complete dominance in case of blood groups? That means A and B genes are dominant over O. So if the gene A exists with gene O, the product will be blood group A. Why? Because A dominates over O. There is a complete dominance between A and O. What about the second case, and B and O? The second genetic case is a lack of dominance. Lack of dominance, يعني there is no dominancy between A and B. يعني A and B, they are strong enough to express itself, so there is a lack of dominance between A and B, so another blood type will be produced, which is AB. What about the third genetic case? Third genetic case is the multiple alleles. As we know from our previous studies that each trait is controlled by 2 genes. In case of blood groups, traits are controlled by 3 different genes. They are, or alleles, they are A, B, and O. But each individual inherits only 2 from these 3 alleles, or from these 3 genes. Only 2 traits will be inherited. Okay? One from the mother, one from the father, as usual. Yani, we have 3 alleles or 3 genes controlling the inheritance of blood groups, but each individual will inherit 2 alleles only, or 2 genes only, one from the mother and one from the father. So, according to these different genetic cases, we have 4 phenotypes for blood groups and 6 genotypes for blood groups. For blood group A, we have 2 genotypes, AA and AO. For blood group B, we have 2 genotypes, BB and BO. For blood group AB, it's only 1 genotype. For blood group O, OO, it's only 1 genotype. This is the genetic classification of blood groups. What about the chemical classification of blood groups? Chemical classification means that blood groups consists of or controlled by 2 chemical substances. They are called, mainly they are 2 different types of proteins, antigens and antibodies. Antigens are they are the proteins that are expressed on the surface of red blood cells, while antibodies, they are in the blood plasma. We have 2 different types of antigens and 2 different types of antibodies. The 2 different types of antigens are antigen A and antigen B. The 2 different types of antibodies are anti-A and anti. Each blood group from the 4 blood groups has its own antigens and antibodies. So let's start by blood group A. It has antigen A and antibody B. Blood group B, it has antigen B and antibody A. Blood group AB, it has two antigens, A and B, but there is no antibodies. Blood group O, no antigens, but it has the two antibodies, anti-B and anti-A. But what is the importance of this chemical classification of blood groups? It's necessary in the blood transfusion. So we need to know the antigens and antibodies of each blood group, which controls the blood transfusion. So let's start to discuss. Firstly, it's not logic to put the antigen with its antibody. Antigen A with antibody A leads to a chemical reaction. This reaction leads to disintegration of red blood cells or breaking down of red blood cells that leads to severe anemia, then leads to death. So antigens and antibodies must not put together in the same individual to prevent or to avoid the chemical reaction that will take place between these two chemical substances, antigens and antibodies. Let's start to explain this part in more details. If we need to transfer a blood from an individual, his blood group A, to another individual, his blood group B, the individual that has a with blood group A has antigen A, and the individual with blood group B has anti-A. So when transferring blood from A to B, antigen A will react with the antibody A. This reaction leads to blood disintegration, breaking down of red blood cells. Severe anemia leads to death. So blood group A, or the person or the individual with blood group A will never transfuse a blood to the individual with blood group B and vice versa. What about the blood group AB? If we are going to transfer blood from an individual A to another one AB, blood A has antigen A, but blood AB has no antibodies, so there is no problem. There is no reaction will take place, so it's safe to transfer blood from blood group A to AB. What about B? The same case, there is no antibodies to react with this antigen, so it's safe to transfer blood from B to AB. But what about the opposite? Can we transfer blood from AB to A or B? Never, because the antigens will react with the antibodies. But what about blood group O? Blood group O has no antigens, but it has the two antibodies. So if we need to transfer blood from A to O, antigen A will react with anti-A. From B to O, antigen B will react with antibody B. From AB to O, antigens A and B will react with these two antibodies. So blood group O will never receive blood from any other blood type. It will receive the blood from only blood group O. What about donation? Blood group O has no antigens, so it's safe to transfer blood group O to A and B and AB. So blood group O is considered as the universal donor. Why it's considered as a universal donor? Because it can transfer the blood to all the blood groups due to the absence of antigens. What about blood group AB? Blood group AB is considered as a universal receiver or recipient. Why? Because it can receive the blood from all the other blood groups. Okay? Now, let's discuss together how can we determine our blood groups. Determination of blood groups mainly depending on the reaction between antigen and antibodies. To determine the blood group, we need two sera, serum anti-A, serum anti-B, and two blood groups from the individual. from the person which we need to determine his blood group. And then we will put in the first blood drop anti-A and the second blood drop anti-B. In our first case, the reaction takes place between the first blood drop and anti-A. So this blood drop contains antigen A, which reacts with the antibody A, leads to blood disintegration. So this blood group is blood group A. So what about the second sample? In the second sample, the reaction takes place by adding the anti-B serum. Anti-B will react with antigen B. Which blood group has antigen B? It's blood group B. And there is no reaction, negative result in the second sample. As in this one, there is no reaction. But when we put anti-A and anti-B in the two samples and there is a reaction takes place, blood disintegration takes place. Anti-B means reaction takes place between anti-B and antigen B, and here anti-A with the antigen A. Which blood group contains the two antigens? It's AB. So this blood group is AB. Here, there is no reaction takes place by adding the two sera for the two blood groups. Why? Because our blood group is O, which doesn't contain any antigens. And this is our method to determine blood groups. Now, we are going to talk about the rhesus factor, or our new antigen. Rhesus factor is discovered in the rhesus species. It's species of monkeys. It was firstly discovered in this species of monkeys, which is called rhesus. So this factor or this antigen is called rhesus factor. This is another antigen beside antigen A and antigen B. So it exists on the surface of RBCs with the other antigens. This Rh factor or this rhesus factor, its inheritance is controlled by 3 pairs of genes or 6 genes. 3 pairs of genes or 6 genes. So to be a positive Rh person, you need to inherit at least only one dominant gene from the 6 genes. Yani, for example, A capital, B capital, B capital, C capital, C capital. These 6 genes are dominant genes. So this individual will be positive Rh. Here we have only 4 dominant genes. You will be a positive Rh. Here we have 2 dominant genes. The individual will be a positive Rh. And if we have only 1 dominant gene, the individual will be positive Rh. When the individual has the 6 genes recessive, the individual will be negative Rh. And about 85% from the human beings are Rh positive and 15% only are Rh negative. This factor is necessary in blood transfusion. So we need to determine the Rh factor and keep it in consideration while transferring blood from a person, from an individual to another. So we have now 8 different blood groups according to the presence and the absence of Rh factor. If the A group has antigen Rh, it will be A positive. So the antigens in case of A positive will be A and antigen Rh. A negative means that this blood group with no Rh. So it has only antigen A, as usual. What about B positive? B antigen and Rh. B negative, B antigen only. AB positive, and this one, which has the largest number of antigens. It has A, B, and Rh. While AB negative, only A and B antigens. O with no antigens, but if it's O positive, that means that this blood group has antigen Rh. O negative, no antigens. And there is no change in our antibodies because at normal conditions, there is no anti-RH. But if we transfer blood from a positive RH person to a negative RH1, the body will deal with the RH antigen as it's a foreign body and it will form anti-RH. And what will happen when the antigen exists with its antibody? A reaction takes place, leads to blood disintegration, which leads to death. So no negative blood group can transfer blood to a positive one. Sorry, no positive transfer to the negative, but the opposite will occur with no problems. So here, our universal donor will be O negative only. Why? Because O positive has RH. O positive maybe donate the blood to the positives only, the positive groups only, but not to the negatives, because if it donate the blood to the negative groups, or anti-RH will be formed. But what is the importance of RH factor to the pregnant woman? Firstly, if the mother is RH positive and the father is RH positive, the baby will be RH positive, and there is no problem. If the mother is RH negative, father is RH negative, the baby will be RH negative. There is no problem. If the mother is RH positive and the father is RH negative, the baby will be RH positive like his mother, because RH positive is the dominant gene. It will dominate over the negative, and the baby will be positive like his mother, and there is no problem. But if the mother is RH negative and the father is RH positive, the baby will be like his father, because his father carrying the dominant gene, but it's not like his mother. His mother is RH negative. And this is a big problem. Why? Because while laboring, during delivery, some of the baby blood, which is RH positive, will leakage to his mother's blood, which is RH negative. And now the immunity system of the mother will be stimulated to produce antibodies against this positive blood. But these antibodies will be formed within 72 hours from laboring, so the first baby will be safe. But what about the second baby? The second baby will be RH positive like his father, and the mother is RH negative, and her body has anti-RH. If the mother is pregnant in the second baby, anti-RH will attack the RH antigens and the baby will die. What is the solution in this case? After laboring, for the mother whose RH is negative and the father is RH is positive, after the first laboring, the first baby, the mother must take a protective serum. Why? To break down the positive RH blood, which is transferred from the baby to the mother before forming anti-RH. So the second baby will be delivered safely. Okay? And this is our fourth non-Mendelian case, which is the inheritance of blood groups. Now, let's talk about genetic diseases. As we know from our previous studies that the normal number of chromosomes in the human somatic cells is 46. Any disorder in this number, by increase or by decrease, leads to an abnormal case. We have about more than 700 abnormal cases, but in our syllabus, we are going to discuss only 3 abnormalities, which are Klinefelter syndrome, Turner syndrome, and Down syndrome or Mongolism. What is the reason for formation of Klinefelter syndrome? Klinefelter syndrome or Klinefelter's individuals are produced due to a disorder that takes place while or during the formation of gametes, which are ovum and sperm. Sometimes during meiosis, the ovum or the chromosomal structure of the ovum will be 22 plus XX, and this is an abnormal ovum, while the sperm is 22 plus Y. What will happen if this abnormal ovum combined by this normal sperm? The chromosomal structure or the product will be 44 plus XXY, and this is an abnormal case, which is called Klinefelter. In this case, how many chromosomes in the somatic cell? 44, 5, 6, 7. Yani, it's not 46. The disorder here is by increase, by 47 chromosomes. The defect is in the somatic chromosomes or the sex chromosomes? The defect is in the sex chromosomes because the normal male has only XY chromosome. Here we have an extra X chromosome, so this case is considered as an abnormal case. What about Turner syndrome? This syndrome is produced as a result of combination between an abnormal gamete, which is 22 plus 0, there is no sex chromosomes, due to a disturbance takes place while the formation of gametes, mainly during meiosis, by a normal gamete, yani 22 plus X. The chromosomal structure, which is produced from the combination between these two gametes, will be 44 plus X, 0. How many chromosomes here? 45. So the disorder is by decrease. The defect in the sex chromosomes or in the somatic chromosomes, it's in the sex chromosomes. Here, can you guess what is the gender of this individual? XXY. Presence of Y chromosome means that this individual is a male. Absence of Y chromosome means that the individual will be a female. So here, there is no Y chromosome, so this case is a female. What about Down syndrome or Mangolism? Down syndrome is produced from the fertilization between an abnormal gametes, sperm or ovum, to produce 45 plus XY chromosomes. 45 plus XY or 45 plus XX, yani disorder is in the somatic chromosomes, not in the sex chromosomes. Defect. Disorder is by increase because we have extra chromosome, 45, 46, 47. But here, males and females, sexually, they are normal, and the disorder is in the somatic chromosomes, especially mainly in the chromosome number 21. This chromosome carries 3 copies of chromosomes instead of 2. Let's discuss the karyotype for each case. Here we have normal autosomes, but chromosome number 23 carrying 2 X chromosomes and Y chromosomes. So this case is a Klinefelter syndrome. Here, normal 22 pairs, but the last pair or the sex chromosome or the pair number 23 with only 1 X chromosome, so we are in front of the Turner syndrome. What about the Down syndrome? We have a normal male, XY, we have normal sex chromosomes, but the defect is in chromosome number 21. 3 copies instead of 2. What are the symptoms for each case? Klinefelter, they are sterile due to the absence of sperm-generating cells, small tests, appearance of some feminine characteristics, طبعا due to the presence of extra X chromosome, mental retardation, extra tall or tallness, growth of limbs more than the normal. In case of Turner syndrome, it doesn't reach the puberty due to the lack of X chromosomes, so which leads to the lack of hormones insufficient. hormones, slow mental growth, presence of some congenital defects in the hearts and kidneys. What about Down syndrome symptoms? Mental or growth retardation, retarded growth, oval face, flat back of the head, shortness, fingers and toes are short, small ears and convex eyes, and they have a mental